Okay, good morning, everybody. My name is Rhonda Henshaw-Powell. I am your moderator for today, and I'm excited to welcome you to this month's technical support webinar series for the ACD support team. And we have an exciting presentation for you from Dr. Tapan Shah. He's going to impress to you how to visualize your RNA and RNA protein co-detection using RNAscope Multiplex Fluorescent Assays. So with that, I'd like to hand over to Tapan. Thank you very much.
Thank you, Rhonda. Could you please confirm that you can hear me fine?
Yes, we can hear you well. Thank you very much.
Thank you, and thank you so much for the kind introduction. So welcome everybody to today's webinar. As Rhonda mentioned, we are going to focus today on how can you visualize your RNA and RNA protein co-detection using the RNAscope Multiplex Fluorescent Assay. Before we get in, I just wanted to give you an overview of the agenda for today. So we'll start with the overview of the technology, followed by looking at the assay workflow, some guidelines on how can you perform successful RNAscope assays, and then a brief introduction into performing the RNAscope Multiplex Fluorescent Assay on the Leica BondRx automation system. And then we'll dive into our newest offering and something that's really exciting, which is the integrated co-detection or ICW workflow, which allows for RNA and protein detection simultaneously. I'll present some of the examples of RNAscope in immunofluorescent staining using the new workflow, provide some guidelines for quantification, and then we'll end with some of the frequently asked questions. So we'll dive right into it, and we'll start with the technology introduction. So as most of you may already be aware, RNAscope is an in situ hybridization method which allows you to detect RNA biomarkers in cells and tissues with a morphological context. It's a very specific and sensitive method because it's got a signal amplification system which allows you to boost signal detection. And with that, it also has a unique probe design, something that we'll look into in some of the slides coming forward. And it allows you to detect single RNA molecule, and you can also detect multiple RNA markers or RNA markers, and you can detect them as fluorescent or chromogenic labeling. And last but not the least, the technology is compatible with a variety of sample types such as FFP, fresh frozen or fixed frozen tissues, just to name a few. So the overview of the workflow is provided on this slide. So the workflow starts with a pretreatment step, which is essential to unmask a target RNA and involves permeabilization of the cells. Once the unmasking of the RNA or the pretreatment has been done, this will be followed by binding of target specific double-Z probes to your target RNA molecules, followed by building of an amplification tree, which is basically sequential hybridization of amplifiers. And then finally, you can detect the signal from single RNA molecules, which can be visualized under a microscope or using a scanner. And ultimately, you can quantify single cell expression from the single RNA molecules that are visualized. So this is basically the overview of the workflow. The unique part, as I mentioned on the introductory slide for the RNAscope workflow, is its custom probe design. So depicted on this slide is the schematic for the probe design. So as you can see, the probe consists of two Zs or double Zs. The bottom part of the double Z is what is specific to your target or is complementary to your target transcript and is going to bind to basically your target region of the mRNA. And it's about 50 base in region. The bottom part is separated from a top part by a linker or a spacer. And the top part of the double Z constitutes the base for the amplification structure, and it's called the pre-amplifier site, which is about 28 nucleotides in length, and this is where your amplification tree is going to be built. So a regular RNAscope probe or target probe set would constitute of about 20 such double Z pairs, or in other words, it will have an RNA target region of about 1 kb or 1000 base pair in length. So the next thing, which is important, I would say, for every molecular detection technology is it's got to be specific as well as sensitive. So the RNAscope technology is sensitive, and the reason I say that is if we go back to the workflow, you have the double Z's binding to your target sequence, followed by the preamplifier binding to the top of the double Z probe, followed by binding of amplifiers to the preamplifier, forming the whole amplification tree, and then detection using an enzyme or a fluorophore. So this amplification system or the amplification boost is what provides the sensitivity to the technology. The technology is also specific, and by that what I mean is if you had a situation where only one of the two Z's bound to your target sequence, the amplification tree will not be formed, and therefore you would not detect any visible signal. So this part of the technology contributes to its specificity. So overall, the technology as required by molecular detection technologies is sensitive as well as specific in nature. Next, I wanted to dive in a little in sharing some of the details about the probe channel nomenclature. And this is particularly important when you're trying to look at multiple RNA biomarkers at the same time. So the way it works is that the double Z probe design has a tail sequence at the top, which indicates a channel designator. So shown here is the tail sequence for a channel 1 probe. The point to remember is, and as you can appreciate from the slide, is that the tail sequence for a channel 2 probe is different from the tail sequence for a channel 1 probe. Or in other words, an amplification tree for a channel 1 probe is going to be specific to a channel 1 probe and will not recognize a channel 2 probe and vice versa. So this tail sequence is what contributes to the specificity and avoids cross detection when you're looking at multiple RNA biomarkers at the same time. Now, as far as the technology and its adoption in the scientific community is concerned, RNAscope has been widely used by researchers all around the world, and it's been very well adopted in the scientific community as evidenced by this bar graph. So we had a first publication in 2011, and since then we've got exponential increase in the number of publications that have used the technology to a point where we have it about close to 3,000 publications now. And if you were to break it down by the research focus areas for which the technology is used, here's the snapshot for that. So cancer and neuroscience turn out to be the two major research focus areas where the technology is used, but that does not take away the fact that the technology is applicable to a wide variety of other research areas such as infectious disease. RNAscope has been in a lot of ways at the forefront in helping with the COVID research, but also research areas such as developmental biology or immunotherapy, to name a couple more. So this gives us an idea about the adoption of the technology in the scientific community. Next, I wanted to give you a snapshot of all the product offerings that ACD has. So shown on this table are the RNAscope, BaseScope, and miRNAscope assays that we offer. Now, I do know this table is slightly complex and has got a lot of information, so I'm going to explain it by breaking it down in columns. So we'll start with the first two columns, which is for the RNAscope chromogenic assays. And you have two options there. You can have a singleplex assay where you can have red detection using the DAB, or you can have, I apologize for that, brown detection using the DAB and red using the ALP detection. And then you can have a duplex where you can have green, red, or red, brown. So you can look at one RNA marker or two RNA biomarkers at the same time. Both these assays are compatible on the manual as well as the automated platforms such as Ventana Discovery and Leica RX. With regards to the compatibility with the integrated co-detection workflow, which allows you to do simultaneous RNA and protein co-detection, something we'll look at in the later half of the talk, only the 2.5 HD red assay is compatible with that workflow at this point in time. So that takes care of the singleplex chromogenic assay offerings that we have. The next three columns, column 3, 4, and 5, are going to focus on the fluorescent assays that we have. So we have the RNAscope Fluorescent Multiplex Assay, Multiplex Fluorescent V2 assay, and the Hyplex assay. So the difference between the three is, the one difference is with regards to the plexing or the number of targets that you can look at. So with fluorescent multiplex, you can look at 3 targets. With the multiplex fluorescent, you can look at 4. And with the Hyplex assay, you can look at 12 targets simultaneously. And the other point to also note is with regards to the compatibility. So at this point in time, all these three assays are compatible with the manual platform, or can be done manually, so to speak, but only the multiplex fluorescent assay is available on the automated platform for Leica Born RX. With regards to the ICW compatibility, only the multiplex fluorescent assay, which has the TSA-based technology, is compatible with the integrated code detection workflow for RNA protein code detection. And then the last three columns essentially represent our sister technologies for looking at shorter transcripts. So you have the BaseScope Red and BaseScope Duplex, and both of these are chromogenic in nature. So you can look at 1 or 2 short sequences. With regards to the platform availability, only the singleplex BaseScope Red is compatible with both the manual and the automated platforms, whereas the Duplex is only available as a manual option. And for the ICW, only the BaseScope Red is compatible with the RNA code detection, RNA protein code detection with ICW. And then finally, we have our newest member on the list, the miRNAscope assay, which allows you to detect mature microRNAs, siRNA, and antisense oligonucleotides. At this point in time, you can only detect one one of the transcripts at a time, but it can be done both manually as well as on Leica Born. But more importantly, it's also compatible with the integrated code detection workflow to detect RNA and protein together. So this was basically a snapshot of all the assays that ACD offers. Apart from that, we also have probes that are going to be used for detection. We currently have more than 20,000 probes in the catalog. And the point to remember is we can design probes against any target from any species, and the custom probe can be ready in as little as 2 weeks. Now, as far as the webinar for today is concerned, we are going to primarily focus on the RNAscope Multiplex Fluorescent Assay, which is the TSA-based detection and allows to detect 4 targets simultaneously. And we'll also look at the data for using this RNAscope assay with the ICW workflow for RNA protein code detection. So the next we'll look at is with regards to the assay workflow on how you can perform with regards to the timing of the assay. So with regards to the assay workflow, for the manual option, you have the option of doing it 3 ways. So you can do it in a single day where you will do the sample preparation, pre-treatment, followed by probe hybridization and detection for the RNAscope assay. Or you have 2 options to split it in 2 days, wherein you can stop after the sample pre-treatment and continue with probes and detection on day 2, or you can stop after probe hybridization and continue with the RNAscope detection on day 2. So these are some of the options on stopping points and how you can split your assay into 2 days if needed. Okay, on the next slide, we're now going to look at the assay workflow for the manual multiplex fluorescent V2 assay. Bear in mind, the workflow shown here is for FFPE tissues. However, the workflow is compatible with fixed frozen, fresh frozen, and cultured cells as well. So as I mentioned at the start of the presentation of the webinar, the workflow starts with pre-treatment, which includes bake and deparaffinization for FFPE samples, followed by a hydrogen peroxide step to kill the endogenous peroxidase, which is important because this is an HRP-based detection that we're going to be doing, followed by antigen retrieval using a hot plate or a steamer to reverse the formaldehyde crosslinks that are formed during the process. And then you will have the protease step to digest the extracellular matrix, which is also crucial for the probes to gain access to the target RNA. The next step is going to be incubating with the multiplex probe for looking at multiple RNA targets at the same time. And then you will do a series of AMP steps, AMP1, 2, and 3, which are going to sequentially build the amplification tree for all of your channel probes. Therefore, keep in mind that AMP1, 2, and 3 are mandatory, even though you were just looking at a single channel probe, because these 3 steps are sequentially building the amplification tree for all the channel probes, and failure to do any of these or skipping any of these steps may result in no detection. This will then be followed by detection of individual channel probes. So you will start with HRPC1 for detection of channel 1 probe, followed by incubating with a fluorophore, which could be opal dyes or TSA plus fluorophores. Please note the TSA plus fluorophores are reagents that you would need to purchase from Akoya Biosciences. followed by the HRP blocker step to block the HRP reaction for that particular channel probe. And you would repeat the steps for the second fluorophore or the second channel probe and the third fluorophore or the third channel probe as well. This would then be followed by the DAPI counterstain, and you would mount with prolonged hold and cover slip the slides. Now on this slide, I've shown you the workflow for only 3 fluorophores or 3 targets, but you can also add on a 4th target to do a 4-target simultaneous detection. In that case, you would need to use the opal dice for detection and not the TSA plus fluorophores. The other thing to also keep a note of, which is a unique feature of these assays, is that you can mix and match the fluorophores, or in other words, the fluorophores are not directly tagged or conjugated to the channel probe. So you could switch the opal 520 in this case, or the opal 570 or 690 for that matter. Okay, so this is with regards to the manual assay workflow. Now we'll look at some of the tips or guidance on how can you get started. First and foremost thing that is required is you will need the reagent kits from ACD. So you will need the pretreatment reagents, which would include target retrieval and H2O2 and protease for the pretreatment steps. Then you would need the detection kit, which is important for the detection part of the assay. And then finally, you would need wash buffer for the washing steps. Please note, wash buffer oftentimes contains precipitate. Therefore, we recommend that you warm the 50x wash buffer for 20 minutes at 40 degrees Celsius before use. And also another point to note is if you're doing a 4-plex detection, you would also need the RNAscope 4-plex ancillary kit for detecting the 4th target. So this is with regards to the reagent kits that you require from ACD. Next, we'll look at some of the recommended accessories that's going to be required for the assay. So you would need the easy batch slide tray and the rack for the slide processing. You will need the Hybridization oven that's provided from ACD, which is critical for maintaining optimal temperature and humidity. And both these parameters are critical to the success of the assay. You would need the tissue tech washing tray for washing your slides. And finally, you would need either a hot plate or a steamer to perform the target retrieval step. The other thing that is also important is that you review the checklist before you get started. I'll go over a couple of the important points on the checklist. One of them being, please make sure that you're using SuperFrost Plus slides. These are chart slides and they help ensure that the tissue stays adhered to the slides during the assay. Use the Hybridization oven and accessory. As I said, the hybridization oven helps maintain optimal temperature and humidity, and both of those are critical to the success of the assay. And then make sure you use the positive and negative control probes, either 3-plex or 4-plex, depending on the assay that you're using, to determine the RNA quality and the background using the negative control probe. The other point that is also important is that you follow the workflow guidelines carefully. Some of the important ones would be, as I mentioned earlier, make sure you perform all the M steps in the right order and do not skip them. Failure to do so will result in no detection or no signal detection. Make sure you don't let your slides dry out unless it is mentioned in the protocol. And then it is recommended that you use fresh reagents such as ethanol, xylene, or 10% formalin all the time. So these are some of the workflow guidelines. And then these are some of the key guidelines overall for performing the assay. So to start with, please make sure that you follow the protocols as exactly described in the user manual. Review the sample pre-treatment conditions that is applicable to your sample type. The sample pre-treatment differs from sample to sample type, and we'll look at that in one of the slides going forward. Ensure to use control probes that are species specific to determine RNA quality. And then, as I mentioned earlier, make sure you review the checklist before you begin to ensure that you have all the materials that are required. And for more information, you may contact ACD technical support at the email address at the bottom of this slide. We'll next look at some of the guidelines on sample preparation. So on this slide, we're looking at sample preparation guideline for formalin fixed paraffin embedded samples or FFPE samples. For FFPE samples, we recommend fixing them in 10% neutral buffer formalin for 16 to 32 hours, at room temperature, followed by standard sample processing, in which you would dehydrate the sample, followed by xylene incubation and paraffin embedding. And then for sectioning, we recommend you cut the sections at 5 plus and minus 1 microns and mount them onto Superfrost Plus slides. With regards to the storage, you can store these sections with desiccants at 4C or room temperature up to 3 months. If you're not going to use the samples right away, then we would prefer that you would store them as blocks rather than sections. And for long-term storage, we suggest storing them at 4C with a desiccant to provide stability to the RNA quality, or in other words, preserve the RNA quality. So this is with the sample preparation guidelines for FFPE samples. Next, we'll look at fixed frozen. So for fixed frozen samples, we recommend that you fix the samples in 4% paraformaldehyde at 4 degrees Celsius for 24 hours, followed by OCT embedding. And then you can store the blocks in -80 for up to 3 months in an airtight container. For sectioning, for fixed frozen, we recommend section thickness of 7 to 15 micron onto Superfrost Plus slides. Please ensure that you air dry the slides at -20 for 1 to 2 hours before you move them to -80, where it can be stored in an airtight container for up to 3 months. I would like to point out that the workflow is also compatible with fresh frozen and cultured cells. I'm not showing the sample preparation guidelines for those in the interest of the time, but it is compatible with those 2 sample types as well. Now, we'll now look at some of the sample pretreatment recommendations. As I mentioned earlier, the sample pretreatment varies from sample to sample type. We're looking at sample pretreatment recommendations for the manual assays. So for FFPE samples, we recommend a target retrieval for 15 minutes at 100 degrees Celsius and using Protease Plus for the protease. Fixed frozen, because they are fixed at 4 degrees Celsius, the target retrieval time is shorter, which is 5 minutes, but you use a stronger protease, which is Protease 3. And then for fresh frozen, since they do not get any fixation, target retrieval is not required, but you would use a harsher protease, which is Protease 4. And finally, for cultured cells, again, since they're not fixed overnight or fixed for too long, they do not need target retrieval, but they would need Protease 3 with an appropriate dilution, because cultured cells, unlike tissues, do not have a lot of extracellular matrix, which requires digestion using the protease enzyme. So these were some of the sample pretreatment recommendations. We'll next look at how do you qualify your samples using the controls, and this is an ideal workflow that we would recommend. So to start with, we would recommend that you perform a technique check using control cell pellets such as HeLa or Mouse 3T3 that is available from ACD. So you run the multiplex negative and positive control probe on those, and you want to see really good staining for all the 3-channel probes or 4-channel probes for the positive control, and clean imaging for the negative control probe. Once you do the technique check, then the next thing you would do is you would go on to do the sample RNA quality check using the same multiplex negative and positive control probes. And once that passes, you can go on to run your target probes. However, if that fails, we recommend that you go back and verify your technique. If the RNA quality in your samples is undermined, then we recommend to check RNA quality with newer samples, and if needed, contact technical support on advice on how can you perform assay optimization for your sample type. Shown on this slide is a beautiful 4-plex staining using the multiplex Fluorescent V2 assay. So we're looking at 4 targets in this case, which is VGAT, CAPTH, VGLUT1, and VGLUT2, and DAPI is a contrast stain. In this case, we're looking at the imaging for fresh frozen mouse brain coronal section. I would like to remind you all again that for the mounting media, we recommend using Prolonged Gold Anti-Fade mounting media. The recommended fluorophore dilution to start with is 1 to 1500, and please remember that TSA plus fluorophores or opal dyes will have to be purchased from Acquire Biosciences for use with this assay. So with this, we looked at the workflow for the manual multiplex fluorescent assay. We'll now switch gears slightly and look at the workflow on the automated platform, which in this case is the Leica Bond RX. So shown here is a pictorial representation of the workflow. It remains very similar to the manual part of it, wherein you have the tissue permeabilization followed by hybridization series for probes and amplification, and then visualization and quantification. We would recommend that you contact ACD sales and Leica sales to schedule RNAscope training and software updates. And I would like to let you all know that we have field application scientists that are available for training and helping you with troubleshooting your assay on the Leica Bond RX. Okay, with regards to the product configuration, you have the reagents that are from ACD, which would be for the LS Multiplex Fluorescent Reagent Kit. You would have bottles in the kit that would have the respective reagents, which have to be filled into the containers or consumables that need to be purchased from Leica. So as I said, you must transfer reagents from ACD bottles into Leica containers. They do not require warm-up before use, and they can only be used with the Leica system. The other thing also to keep a note of is that the bond research detection kit and the containers must be purchased or obtained from Leica Biosystems. With regards to the workflow or how much can you do with a single kit, the LS Multiplex Assay provides enough reagents to stain 60 standard slides. However, each probe for the Leica Bond platform is sufficient for staining 30 slides. The runtime is roughly 14 hours. And as I mentioned, you would need to get the bond research detection kit from Leica, which is required to set up the LS Multiplex Fluorescent Assay. The signal detection will be performed using opal dyes or TSA plus fluorophores that have to be purchased from Akoya Biosciences. And like the manual option, if you wanted to do the 4-plex detection, you would need the 4-plex Leica ancillary pack to detect the 4 targets. And for the 4-plex assay, you would need to use opal dyes. Okay. Now here's a software interface snapshot for the Leica Bond RX. We are looking at the interface for the software 6.0 version. So at the top, you have options for setting up your slides, protocol setup, and reagent setup. On the left, you have the option of toggling between 2 instruments if they are connected to the controller. And then you have the SSA 1, 2, and 3, which is where your slide tray will go into. And then this is where you would have the reagent trays with all these solutions. And then at the bottom here, you have the representation of the bulk that are on the instrument. Okay. And then we'll now look at some of the sample pretreatment guidelines for the Leica Bond RX platform. So for FFPE samples on Leica Bond, we recommend doing 15 minutes at 95 degrees Celsius with ER2 buffer for tissues and 15 minutes at 88 degrees for cell pellets. And the protease will be protease 3. For fixed frozen, you would do 5 minutes, just like the manual version, and protease 3 for the protease enzyme. And then fresh frozen, again, since it does not require any target retrieval because of no fixation, it will get a stronger protease, which is protease 4. And cultured cells, like the manual option, do not require ER2 or target retrieval, but will use protease 3 with an appropriate dilution. On this slide is shown an example of a 4-plex staining using the LS Multiplex Fluorescent Assay. In this case, we are looking at immune cell infiltration for non-small cell lung cancer tumors, and these are FFPE sample types. In this case, we're looking at CD8, which is a marker for cytotoxic T cells, CXCL9, interferon gamma, and CCL5. And the blue arrow is indicating a cell that is positive for all the 4 targets, so CCL5, CXCL9, interferon gamma, and CD8 positive. Okay. Now, there might be situations where you would need to troubleshoot your assay, and the question might be, why do you need to optimize your assay? So oftentimes you have situations where you would have underfixed samples or overfixed samples, or you might be working with other sample types which may require a little extra care, such as xenograft or culture cells, because culture cells you will need to determine the protease dilution that is appropriate for your cells, working with whole mount tissues or cell pellets, or tissues such as liver, muscle, or retina, so to speak. In the interest of time, I will not be discussing the troubleshooting guidance for the webinar today, but you can refer to the previously recorded webinar using the link at the bottom for troubleshooting tips for fluorescent assay. So this was with regards to the Leica Bond RX platform for the fluorescent assay, and then some of the troubleshooting guidance. We'll now switch gears and look into the exciting new product that we have available, which is the integrated co-detection workflow for simultaneous RNA and protein detection. So to start with, I wanted to give you an overview of the sequential dual RNAscope ISH-IHC workflow that existed before the ICW. So the way the previous workflow worked is you would permeabilize the tissue, followed by hybridization of the probes. You amplify the RNAscope signal, and then you go on to bind your primary antibody to the secondary antibody and then visualize the morphology. The caveat with this workflow was the fact that during the pretreatment step for RNAscope, you have the protease enzyme, which may negatively impact the protein expression of your target gene or the target protein that you're trying to probe using IHC or IF. And therefore, to get around that, we came up with the new integrated co-detection workflow. And as you can see here, the process starts with permeabilization, where you would do only the antigen retrieval step. And this will then be followed by binding of your primary antibody to the protein target of your interest. And you would then go on to do the hydrogen peroxide and the protease step, followed by the probe hybridization, amplification, and detection of the RNAscope signal. And then you will have the secondary antibody detection, and you would visualize the RNA-Protein Detection simultaneously. So the biggest advantage, as I said, is the fact that you have your primary antibody bound to the protein target before you add the protease, which may be otherwise detrimental. And therefore, this workflow enables use of more antibodies with the RNA protein co-detection. It also reduces the need for extensive screening for compatible antibody clones. So therefore, you would have faster time to results. The performance is equal or better with regards to antibodies that are compatible with the sequential co-detection. And it's got equivalent sensitivity to ISH-only detection and more faithfully retains the specificity of the antibody or IHC alone. So these are some of the advantages of the new integrated co-detection workflow, or the ICW. With regards to the compatibility and the products that you would need for the kit, you would need the co-detection blocker and the antibody diluent, which is also sold as a standalone product. And you would need the co-detection target retrieval reagent. Now, the thing to remember is that the blocker and the co-detection target retrieval are applicable to manual assay only. And for the Leica bond platform, you would still use the epitope retrieval 2 that is on the instrument. With regards to the compatibility, as I mentioned in the slide earlier, which snapshotted all the assays that we offer, the ICW is compatible with the multiplex Fluorescent V2 assay. It's compatible with the BaseScope and RNAscope Red assay, miRNAscope assay, and on the Leica bond platform, it's compatible with 2.5 HD Red, LS Multiplex Fluorescent assay, which is what we're focusing on today, BaseScope Red assay, and miRNAscope Red assay on the Leica bond RX. Okay, now shown on this diagram is a schematic for the co-detection workflow. This shows the workflow for both manual and the automated versions, and I'll point out the points of difference along the way. On this slide, we've shown the FFPE sample type workflow, but it's also compatible with fixed and fresh frozen tissues as well as cultured cells. So the procedure starts with the pre-treatment step, where you would do bake and dewax for FFPE samples, followed by antigen retrieval, and you will then do the primary antibody incubation and a post-primary antibody fix. The thing to note or keep in mind is for the manual assay, the primary antibody incubation is overnight at 4 degrees Celsius, whereas for the Leica bond, it's going to be 15 minutes at room temperature. Once you've done the primary antibody incubation and post-fix, you would then do the protease step to chew the extracellular matrix, followed by peroxidase block to basically block the endogenous peroxidase activity. And then you would do the multiplex probe hybridization, build the amplification tree for RNAscope probe with AMP 1, 2, and 3. And then you would do the multiplex co-detection part A, which is going to be for RNAscope. So you will do the detection for channel 1, which is HRPC1, followed by TSA reagent 1, which can be TSA plus fluorophore or opal dyes, followed by the blocker step. And then you will repeat this for channel 2 and channel 3. And after that, you will go to the part B of the workflow, for which you have 2 different options. For the IF detection, so you can do the detection using the opal dyes, like you do the RNAscope assay, which would involve like a post-primary and like a polymer HRP on the Leica bond, and HRP conjugated secondary antibody for the manual assay. This would then be followed by using a TSA reagent, so an opal dye or a TSA plus fluorophore, to detect the HIP conjugated secondary antibody, followed by DAPI and mounting with prolonged gold and cover slipping. That's the first option for the immunofluorescence detection. The option 2 is where you can use a fluorophore conjugated antibody or fluorophore conjugated secondary antibody, which means you do not need to use the TSA reagent for detection. So you would just use the conjugated antibody followed by the DAPI counterstain. You can mount with prolonged gold and cover slip. So this is the schematic for the ICW workflow for the multiplex fluorescent assay. We'll next look at some of the images or staining results for the RNA protein co-detection using the new ICW workflow. So shown on this slide is an example on how the ICW workflow improves dual RNA protein detection. So on this slide on the top panel, you have dual staining for RNA protein. So we are looking at human PPIB and CD3 antibody in the top panel, and in the bottom panel we have human PPIB for RNAscope and CD8 antibody for the IF in lung cancer FFPE samples. So in the first panel you're seeing the IF alone, so this is immunofluorescence alone, and as you can see the striking difference when you look at ISH-IF without the co-detection reagents, and when you compare it with the ISH-IF with co-detection reagents, there is a striking difference. And more importantly, if you look at the detection, the ISH-IF with co-detection reagents for the immunostaining for CD3 and CD8 are very compatible to the IF alone, suggesting that the ICW will help improve the dual RNA protein detection. Shown on this slide is an example of a 3-plex RNAscope ISH with an antibody staining for Ki67. So on this slide, we are looking at polar 2A in red, PPIB in magenta, and UBC in green. And then in white, we have the Ki67 for cell proliferation marker. We'll next look at some of the examples for immune markers. So on this slide, we are looking at, again, 3-plex RNAscope ISH with CD3 antibodies. So here you have CD3 in green for RNAscope, PDL1 in red for RNAscope, and CD8 in yellow for RNAscope with CD3 in IF. And the box here, which is expanded here, shows co-detection in green for CD3 RNA and CD3 IF in white. So this basically depicts the T cells which are positive for CD3 RNA and protein in FFPE human tonsil sample. We also have a switched situation wherein we now have the same probe combination for the RNAscope assay, but now we're looking at CD8 for IF. So what you have is you have your general T cell population, which is positive for CD3 RNA, but now you have your subset of T cell population, which in this case is cytotoxic T cells, which is positive for CD3 RNA, which is for the general T cell marker, but it's also positive for CD8 RNA and protein, which marks the cytotoxic T cells in yellow and white respectively. Okay, and then the final example I wanted to show you is with a tumor sample. So we are looking at FFPE human breast cancer TMA in this case. And what we're looking at is we're looking at the same markers again with CD3 antibody. And on the slide, you see a nice demarcation of the stroma here, separated from the tumor, which has got the PDL1 positive cells in red. But what you're also seeing in yellow box is potential tumor T cell infiltration, which is depicted by CD3 and CD8 RNA positive and CD3 IF positive cells. Okay, so these were some of the examples for the multiplex RNAscope staining with immunofluorescence using the new integrated co-detection workflow. We'll next look at some of the guidelines on how you can quantify the fluorescent signal. So some of the key questions with regards to assay quantification for the RNAscope assay are: what does a dot mean? What is the significance of a dot size? And what is the difference between a dot and a cluster? And the answer to all these three questions will be depicted on the next slide. So in this case, you're seeing different scenarios for signal detection. The thing to remember is that all mRNA molecules with RNAscope are detected as punctate dots, and each dot represents a single copy of the mRNA or the transcript. So in this case, you have single dots that you can see with yellow arrows, or the white arrows, so to speak. What you can also have is you can have clusters that would overlap from signal from mRNA molecules that are very adjacent to each other, shown in yellow here. However, what you can also have, is variation in dot size and intensity, which is related to difference in the number of ZZ pairs that are bound to the target molecule. So in a scenario where you have more ZZ pairs that are bound, the dot size would be brighter or stronger, as shown here, as opposed to a scenario where you would potentially have less number of ZZ pairs bound. But the thing to remember, as I mentioned at the start, is that each dot represents a single transcript, and it's the number of dots that is important and not the size of the intensity of the dots. With regards to the scoring guideline, we have two options. You can do the semi-quantitative scoring as shown on the slide here. And the scoring basically has like 5 different bins where you have a score of 0 to 4. 0 means no staining or less than 1 dot per 10 cells, which increases to a score of 4 where you have more than 15 dots per cell and more than 10% of the dots are in clusters. And a pictorial representation of a score 0, 1, 2, 3, and 4 is shown in this case. Okay, the quantification can also be done in a more quantitative method. So for that, you can use open source image analysis software such as ImageJ or from Fiji or Cell Profiler, and using those two open source softwares, you can represent the quantified data as total cell count, the total probe dot count, average number of dots per cell, percentage of positive cells, and percentage of dual or triple positive cells. And here is the link for downloading the ImageJ from Fiji and Cell Profiler for your reference. We also have resources for data analysis that are available for you. So you can have guidelines on how you can quantify the signal, or how to use ImageJ or Cell Profiler. So these are our tech notes for analyzing the RNAscope and BaseScope data. And you can download these resources using the link shown at the bottom of the slide. With that, I come to the end of the webinar today. And I would like to encourage you to contact ACD technical support for all the technical support. We have technical support scientists that are available to help you with your assay, RNAscope as well as BaseScope, and ensure you're not stuck. You can contact us via email using our email address, which is support.acd@bio-techne.com, or you can contact us via phone using the phone number mentioned here. And then going on to option 3, we have support scientists that are available to speak with you from 8 a.m. to 6 p.m. Pacific Standard Time. And then you can visit this link for support resources. So this is with regards to the contact information for technical support. As Rhonda mentioned earlier, we have my colleagues or technical support scientists that are available to answer your questions. So please feel free to put your questions into the question window so that we can have our panelists answer those. And in the next 2 slides, I'm just going to have slides that are going to look at some of the frequently asked questions. And with that, I would like to thank you all once again for attending today's webinar. And I really hope what I presented to you all today was helpful and will help you move your projects forward using our RNAscope technology.
Thank you very much, Tapan. That was an excellent presentation, and thank you all for attending today. We did get a good amount of engagement and questions coming through. So if you didn't get your question answered, we are actively working on them now, and if we close the webinar, then we will reach out to you directly via your email contact information. So please have a look at these frequently asked questions. And if you have any questions about those, pop them in the question top. We will be online for another couple of minutes while we go through these slides. And again, thank you very much for the support team, and thank you to Tapan for an excellent presentation. And one final note, this webinar was recorded, and so hopefully by the end of the week we will have this online. If you go to the acdbio.com and look at the recorded webinars section, in the next couple of days we will have this webinar available for you to rewatch on demand and share amongst your colleagues, so please do that. Here is the second slide of the frequently asked questions. If you have anything else to ask us, please go ahead and do that. If not, enjoy the rest of your day, and we look forward to seeing you in our next series of webinars. The ACD team hosts webinars quite frequently, and you can check them out on our events page. If you go to our main homepage and scroll to the bottom, you'll see a list of events. The support team do give presentations each month, and so please look out for those as well. And if you have any questions at all, reach out to the support team at the address noted here, support.acd@biotechne.com. Also check out our resources. There are a lot of questions today about all areas of RNAscope and everything that we do. So please do check us out, and the tech support team are readily available for you to answer any questions that you may have. Okay. With that, I am going to close today's presentation. Thank you very much. Goodbye.